The first time was some 20 years ago when I established a PhD student with Paula Bramall, who was going to work on the development of core collection of sorghum. The second time was within the Generation Challenge program. We interacted with Harry Upadhyaya on the evaluation of reference sets of sorghum again. This time of the Generation Challenge program gave me the chance to interact with Rajiv. I was leading the genetic diversity sub-program, and he was leading the genomics sub-program. And together we co-authored a paper called More Genomic Resources for Less Studied Crops. And I'm very happy to see today that this paper is completely obsolete, because the less studied crops are no longer less studied, they are champions, and we heard a lot about these crops these days. With this, I'm going to talk about genome harvest, which is an initiative that we have in Montpellier. First, my interest is crop evolution, and given the availability of genome sequences and resequencing data, we can look at the foundation of modern crops. And the idea is that this foundation is usually heterogeneous, given the history of the crops, the domestication, and then the migration with human populations, followed by drift, and then possibly adaptation, and then admixture. The whole populations today, we believe, are like mosaics of ancient diversity patterns. And that's what we want to see with the Genome Harvest project. This project has been funded by Agropolis Foundation. The idea is to decipher mosaic genome diversity patterns as pathways to crop improvement. And this associates people that are very good in statistics, informatics, evolution, population genetics, and on the other side, genetic resources and plant breeding, genome analysis of crops. This project, correct? Oh, yeah, thank you. Has a structure, and I am actually in charge of Work Package 2, that's looking at mosaic genome structure within crop genomes and large structural variation with crop genomes. And we look at different crops: citrus, banana, rice, coffee, and a second round of tomato, groundnut, and sugar cane. And I'm going to talk about rice and banana today. Okay. So the first crop is rice. As you mentioned, kindly, I've been involved in rice since the early days of genetic markers, biochemical markers, and I'm very happy to resume work. Sorry, I can't. Well, that's a nice picture. Can I use this?
Yeah.
Okay. So that's essentially based on lysozymes. The diversity in rice can be summarized with the existence of two main groups: Japonica, with temperate and tropical versions, and Indica, essentially tropical, and a range of special types around the Himalayas, including the Rose type, ecotype from Bangladesh and Northeast India, and the well-known Basmati type that is famous for its aroma. There are other types like the Rayada, that is floating rice, but these are minor in size. So we are fortunate to have the availability of this data that was produced by the 3000 Rice Genomes Project, an initiative between IRI and the Chinese VGI, led to the resequencing with a good depth of these 3000 accessions, with communication and free access to public since 2014. And with the development of a very good informatics environment with the International RISE Informatics Consortium and the SNP-seq database and regular publications on the SNP-seq database with updates. So we are very fortunate to have this great resource, and we are using it. So this shows a summary of the structure revealed with many 100,000 markers, and you can see the three poles are indica, japonica, and we call it C-house for circum-house or containing house, because there are other varieties than house ecotypes, and this one circum-basmati, because there are other varieties than basmati, and some are non-aromatic. We shouldn't call it aromatic, but that's the structure as summarized from all these big amounts of data. And of course, with so many data, we can go into more depth. And if we compare the structure on the left side with the geographic origin on the right-hand side, we can see, I won't go into the details, but we can see that there's a strong geographic structure, especially in the Japonica subgroup, but also in the Indica subgroup, and two different types in the house group. So I need to give you some context, because we have had the most complete study to date. It was published in 5 years ago in Nature with the resequencing of more than 100,000 cultivars and 500 wild accessions with about 5 million SNPs. And these data were analyzed by different groups. First, those who produced the data with this publication. So the wild rice can be summarized with this structure and this geographic distribution. Then, if we look at whole genome accumulating cultivated and wild, we can see that there are two types: the Japonica, with cultivated and wild, and the Indica, with cultivated and wild. So that's fine. We have both the two major types and their wild homologs. But when we look at genomic regions which have a decreased variation when you move from wild to cultivated, that means that may be involved in domestication, we can see that the Indica varieties cluster with the Japonica varieties together with a group of wild rices that are from South China, and that leads to the conclusion that there has been one domestication in southern China, and that these domesticated forms actually enhanced and accelerated the domestication of Indica types. So Indica cultivars cluster with Japonica wild. When you look at these genomic regions with restricted diversity. So another group looked at the same data and selected the regions, more regions with decreased diversity, and actually showed this pattern with Indica, Japonica, but also a group that corresponds to the house accessions. And this led them to conclude that there has been 3 geographically separate domestications in rice. So we can summarize this with these two contrasting figures. On the left, one domestication in Japonica that led to Japonica type, and then that accelerated by introgression of the domestication syndrome genes, acceleration of domestication of Indica and house varieties. That's one hypothesis. The other one is a triple domestication with different separate evolutions. Of course, there has been some admixture here, and there's also a contrast in the interpretation of the origin of the C. basmati rice. Here it's considered as a subgroup of Japonica. Here it's considered as a result of an admixture between house and Japonica. Third group looked at the same data, and to make it short, they could find in the wild genomes traces of introgression from house in this color in India or Bangladesh, from indica in blue in Thailand and Bangladesh, and from japonica in China. That means that while current wild populations are strongly contaminated by cultivated varieties through gene flow, through pollination. That means that makes it difficult to apply simple rationales to try and decipher the domestication of rice. So our work, our approach is to try and look at the diversity of, in these varieties with this representation to start with. The representation we have when we start our work is like this, possibly 2 or 3 domestications with a lot of gene flow between the accessions and including some wild that yields current patterns of genomes like this indica with some introgressions from japonica. It could be the domestication syndrome, it could be other types. Japonica on the other side may have some indica or other introgressed regions, and around the Himalayas, a number of groups, including the C house and the C basmati, but many other patterns as well. We are particularly interested in this data because we want to try and disentangle 2 sources of adaptation. One can be the incorporation and the admixture starting from standing variation that was present early in the domestication and it's present in the wild. The other one is the novel variation that may happen during crop evolution. And if we make counts, the number of plants of rice that are grown every year in the world is so huge that we are sure that any mutation occurs many thousand times in the world every year for any single base pairs of the genome. So it's likely that new mutation can be picked up if they are favorable, can be picked up by farmers. So this part may be not negligible. But if we want to resolve between the two, we'd better understand these patterns of admixture and introgression first. So one major element is the dialogue between what we see in the local, in the genome segment, what we call local structure, and the global structure that we know is japonica, indica, CO, C basmati. So for doing so, we can apply multivariate analysis locally and make use of machine learning tools to apply kernel density estimation so that we can try to automate local classifications. So this shows what we can do with these tools. I have to be quick here, but if the local variation is like this and relates like this to global groups, we can see here that there are incongruities, and here that there may be outliers, and the machine can reclassify and identify outliers and give a result to us the possibility to assign the segments of any accession in any region to one of the poles, japonica, indica, or C house, or maybe intermediate colors if there is some ambiguity, or maybe some ambiguity between 2 would lead to the intermediate color, and intermediate between 3 would lead to the gray color, but possibly there is actually no resemblance between what we see locally and what is the global structure. And in this case, it may be another pole, another foundation that was not present in the initial representation. So with this, we can draw circles and represent the diversity of the genome of a number of accessions. Here, each circle represents one accession of rice, and the colors represent what I just described. This is a sample of about 60 Japonica accessions. And we can see that they are globally blue. 80% of the genome is blue, corresponds to the Japonica group. But there are chunks of other colors, like here was probably introgression of indica materials. Here there is a region that is ambiguous between Japonica and Aus. That could be something that is common to Aus and Japonica and absent in indica. Here, this purple means that this is common between Japonica and indica and absent from Aus. So with this type of representation, we can have a hint on the genomic constitution of all the accessions. And if we looked at accessions from the C-Aus pole, from the Japonica pole, from the indica pole, they have the main color. And in the middle, we have the representation of the C basmati group. And you can see how heterogeneous this is. There are common things common to all the accessions, but there is a huge diversity. Globally, there are 40% of Japonica chunks, segments, 15% of Aus segments, 10% of indica segments, and 15% of segments that correspond to none of these three poles. And that could be related to specific contribution by local wild rices or something that we may interrogate. So with this type of representation, we can go further in exploration. For example, we can look at this region that is homogeneously blue and look in Japonica to what region to what geographic region this genomic region links C. basmati, C. japonica, to understand which part of the Japonica pool can have contributed to the basmati. And same thing with Aus, same thing with indica. So with this, we can explore the origin of the C. basmati group with more method and more rationale. Now we know that we have a different issue. We have different reference sequences. Here, this is aligned to the Japonica reference sequence, but we know that indica and Aus may have slightly different maps with lots of structural rearrangements. We can look at the contrasted epistatic balances. Why did the big program of indica japonica introgression that was run in the 1950s fail? How come? How come there is not more mixture than this? That may be due to some strong interactions between different parts of the genome. So we have a range of questions that are very relevant for breeding. That's the point where we are. We are continuing this study with Joe Santos, the student who is doing this thesis. Now I have another 5 minutes only for banana. Banana, we were fortunate to sequence the genome. We had the front page of Le Monde, which is something good. This is a little sunlight brought by banana to the tourists in France where it was a very rainy summer. And I'm going to talk about the results of this study. So introduction about banana, we have two genomes, Balbizana and Acuminata. I'm focusing on Acuminata here. There are subspecies, and there are regions where these different subspecies are in contact. And that's where this hybridization occurred, which developed, developed seedless parthenocarpic hybrids that you can see here a banana and here a wild banana with the seeds inside. Looking at meiosis, Shepard described 7 structurally homogeneous groups in terms of cytogenetics. Looking at genetic maps and skewed, distorted segregations, we can also have hints on structural rearrangements. And we have focused on a particular case. This shows the segregation of DART-seq markers among 180 individuals in the self-progeny of a particular cultivar. And we can see here the linkage that corresponds to the map. But we can see also here that there is a pseudo-linkage between two different chromosomes. And we can see here that there are skewed distributions, skewed segregations in the regions that correspond to this pseudo-linkage. That's highlighted here. So there is also a region of reduced recombination here. That means, does it relate to structural variation? So we looked at, we used amplification and sequence method to try and pair, so using a made pair Illumina sequencing, we were able to link the two chromosomes here, chromosome 1 and 4, that correspond to the translocation between chromosome 1 and 4. And via one external repeated sequences, we could also link chromosome 1 and 4, the other segments here. So this is a little conjectural, but that's proven, and that works very well with PCR amplification. We can amplify a specific fragment where there is a translocation. And it also works with the backfish, where we could identify, demonstrate the conjectural interpretation that I mentioned earlier. So we can look at diversity with lots of resequencing data. This shows different subspecies, and the cytotypes homogeneous for the alternative structure, the translocation, are highlighted here. They are concentrated in the white in the subspecies malacensis. So the idea is that this structure emerged in malacensis. This is not found in related species, so this reference structure is likely to be the ancestral one. So at the end of this study, we are able, with a simple PCR, to characterize the presence or absence of a particular translocation. That's a really big progress. Now, a few words on the B genome. The B genome is very important because it participates in very important varieties, notably the plantain, that is AAB triploid. And the B genome has two translocations and one inversion compared to the A genome. And the plans for breeding plantain are making crosses using plantain with sources of interesting genes, going through different types of ploidy in order to release triploid progenies. So in this case, we looked at this clone, a tetraploid, expectedly AAB, and using sequencing data and normalizing, using the frequency of the sequences obtained, we could normalize and identify in this particular clone regions that have only the A genome, only the A allele is present, regions that have the same amount of A and B alleles, and regions that are, as expected, triple amount of A versus B. So this produces this, allows us to infer the cytotype of this particular clone on each of these chromosomes. Chromosome constitution is more complex than expected. There are suites of AAA or AAAB or AABB, and there are multiple AB recombinations. So for the sake of time, I will skip the last figure and just insist that with these techniques, for 200 euros today, maybe less tomorrow, we can determine the genomic constitution of a particular clone that is in our breeding programs, derived from crosses between different cytotypes, and where we are not sure how many A or B chromosomes there are, this can be done so quickly. So we can expedite genomic constitution, typing A, B, and euploids, and explore improvement pathways with more efficiency. To conclude, this initiative is actually putting together people from different disciplines, including mathematics, evolution, plant biology, breeding, and we are very happy to have this focus on a diverse crowd, on the same focus, same concern. And I want to finish by thanking the Banana Group, Angélique Dante, Guillaume Martin, Frank-Christophe Borins, Nabila Yayaoui, and my colleagues who also work on RISE with me, Joe Santos and Gal Tondra. And with this, I thank you for your attention.